One of my proudest achievements has to be in setting up the medical entomology team. There was no capability for entomology within UK government when we started. We've been able to identify new species coming into the country and deal with those incidents. We've been able to provide a map of where Ixodes ricinus ticks for Lyme disease are. We established a national tick surveillance / national tick survey with national parks, so we now understand what infection rates are within ticks for Lyme disease. Been part of research work with the virology and pathogenesis team that detected tick-borne encephalitis virus for the first time. We've also been involved in invasive mosquito surveillance, and we set that up and we've detected mosquitoes on 6 occasions, and working with local authorities and health protection teams managed to eradicate those species, so trying to forestall the invasion of these species. And everything that we do is available transparently, so we try and publish all our surveillance data and all our research. So there's a constant process for our team analysing data, writing up papers and publishing that in peer-reviewed papers. We're at the point in some of our research where we're finding sort of ecological drivers and hopefully environmental solutions. Medical entomology is a study of insects that are important in public health, specifically the transmission of infectious diseases. In Northern Europe, the primary concerns for public health are those transmitted by mosquitoes and ticks. So we work specifically on the entomological and ecological aspects of these diseases. And this is all about understanding what the risks are from arthropods to public health. That's understanding where the species are, which species might be involved in transmission, and what their likely risk is to public health in the future. What do I do? Hi, I'm Dr. Jolien Medlock. I'm Head of Medical Entomology at the UK Health Security Agency. The Medical Entomology and Zoonoses Ecology team really is all about insects and the disease transmission scenarios for vector-borne disease. So people who work in the MESE team have a background in vector-borne diseases and maybe in mosquitoes or in ticks or disease ecology or some aspects of transmission related to viruses or bacteria. I started working for the Centre for Applied Microbiology and Research at Porton Down in 2002. And since then I've worked for the Health Protection Agency and Public Health England and now the UK Health Security Agency. The prominence of vector-borne disease over that time has increased. They're now very much more concerned about the climate-related risks from vector-borne diseases. We've seen the emergence of things like West Nile virus, dengue virus in Europe, the increase in Lyme disease, the spread to northern parts of Europe, including the UK, for tick-borne encephalitis virus. So the prominence and the need for medical entomology is only heightened throughout that period. The team has a core function within the organisation, firstly to provide advice to government through our One Health groups, such as the Human Animal Infections Risk Surveillance Group, also through membership of advisory committees such as the Advisory Committee on Dangerous Pathogens, and to our core goals of making sure that we protect the nation's health from arthropod-borne, insect-borne diseases. Our day-to-day job is pretty varied and varies throughout the year. So in the spring, during sort of April, May and June, we're very much focused on managing our tick surveillance scheme. So that's samples being sent in from the public, from GPs, from vets, from wildlife charities. And the peak number of submissions from the public come in around June time.
And what we ask people to do is to fill out a form when they send their samples in to us, to tell us when they found their tick, where they found their tick, and anything else that they want to tell us about their tick encounter. This one's coming from a dog removed in springtime, which, as we know, is our busiest time for tick activity. Our main species that we have in the UK is Ixodes ricinus. This is also called the sheep or deer tick. It's the same species and it's found feeding on pretty much anything. It's not very fussy. It's got a really broad distribution across the UK and it's most commonly found in the sort of spring months, often when we want to be out and enjoying the nice weather as well. We've also got another species that we're interested in called Haemaphysalis punctata. This species has also been found feeding on people, and that's why we're interested in monitoring the distribution of this one. If it does become a future vector of interest, then we'll know exactly where it's found and when it's found, and we can react.
Nice.
Hi, I'm Katie Hansford, and I'm a principal medical entomologist working for the UK Health Security Agency. So my job involves assessing the risk of ticks to UK public health. This can involve running some of our national surveillance schemes and also coordinating some of our research, where we're looking into ecological drivers of pathogens that cause diseases like Lyme disease. This map's showing us where we've had most of our tick surveillance scheme records recorded across the country. And as you can see, they're quite grouped in the southwest, which is where we do see lots of tick activity. They're also showing with each of these local authorities where we're seeing an increase in the number of records that are actually being sent in from these individual areas. So this gives us an indication that these areas might be at a higher risk of tick bites. And what we're doing with this data is working with the Emerging Infections and Zoonoses team to have a look at the Lyme disease incidence data across these local authorities to see if we can link them up. We'll use this to inform our public health risk assessments of tick-borne disease risk at the local level.
Close to Porton Down, we have privileged access to a large woodland ecosystem that we've used now for 15 years, both for long-term surveillance but also for a range of different research projects. What we're doing here is monitoring the density of ticks. By dragging this cloth just over the vegetation, it mimics an animal. The ticks tend to spend their time questing in the vegetation at the tips of the grass, and they're looking for an animal host.
When ticks are at the top of vegetation questing for a host, they're often looking for cues of host activity. And the main tick species that we've got in the UK doesn't have eyes, so it can't see the hosts. It needs to use other methods to detect this activity. And it has something called a Haller's organ on the ends of its front legs. So whilst it's in the grass questing, it'll be waving these legs around, picking up these cues. And the kind of cues it's picking up are CO2, changes in light, and also heat. And this gives them an indication that a host might be nearby.
Here we're calculating the numbers of ticks per unit area, and then we'll take those ticks back to the lab and test them for things like Lyme disease and tick-borne encephalitis virus. This gives us a metric on what proportion of ticks across the country are able to transmit infection to humans.
Okay, so these ticks that we've just collected, these larger ones at the bottom here are adult females, and this smaller group up here, these smaller ones, are nymphs. And when they bite us, it's this nymphal stage that is really important in terms of public health. And that's because their small size means that we sometimes miss them when they've bitten our skin. And the longer that they're attached and feeding, the higher the chances there are of them transmitting the bacteria that can cause Lyme disease. So it's really important when you've been out in glorious woodlands like this to do a tick check when you get home. Once we've collected questing ticks from the field, we bring them back to the medical entomology lab. Firstly, we look at them under the microscope to identify which of the 20 UK tick species we've actually found. They then go off to our PCR lab, where we run a test to look to see if we can find different bacteria and pathogens that might cause disease in humans.
We tested these ticks to see if they were carrying the bacteria causing Lyme disease. And we are going to look at all the ones that are positive, which are the ones that have this curve on the screen. And then we will check for all the samples in the plate. And so, for example, these two are also positive for Borrelia burgdorferi. And then the next step will be to sequence these samples, because different genus species can cause Lyme disease of the genus Borrelia burgdorferi. So we'll use some sequencing method to be able to identify exactly which genus species these ticks are infected with.
There are also a number of universities, very good universities within the UK, that have leading expertise in tropical medicine and vector-borne diseases. And we work with all of those who have an interest in working collaboratively with UKHSA, both on specific projects outside the research councils, also funded by the research councils. And they bring unique expertise that we don't necessarily have within our team.
So in this project, we're trying to understand the drivers of Lyme disease. So we are setting up camera traps in the forest, so we have 100 traps deployed in 30 woodlands, and we rotate them monthly in random locations. And the goal is to take pictures of animals in the forest so we can assess the density of hosts, because some hosts, especially deer, are very important for in driving tick numbers. So in this project, we're really hoping to assess deer density in these woodlands and to relate that to the density of ticks that we're also getting, and then to input all this data into infection disease models to understand what are the drivers of Lyme disease. So we set up the cameras at 40 centimeter height, so which is the height that it will pick up most species of deer. So it's activated by motion, so once it's turned on, when an animal will pass in front of a camera, it will trigger it and will take a picture. So we've had settings that it will take one picture every second. And we have this pole set up so that we can calibrate how far the animals are when they get caught on camera. So we always check that we can see all the poles in the spring so that we know we will detect any animal that will be there. And once it's set up, we always take one picture of just the poles for calibration. Once this is set up, it will be left for a month. Take pictures and we will come and switch the SD cards and then move the cameras around.
Is that for me, is it? Yeah. No way. Hello, I'm Alexander Vaux. I'm a principal medical entomologist in the medical entomology team. I work on both invasive mosquitoes and native mosquitoes in the field. For invasive mosquitoes, I'm particularly interested in Aedes albopictus, the Asian tiger mosquito. Our work on invasive mosquitoes is collaborative with local authorities, particularly who will run mosquito traps in their local areas and send the substrate, the polystyrene blocks, to us for checking in our lab. That includes surveillance at a range of different sites, including service stations on the motorway networks, in particular targeting cross-continental traffic, or other high-risk sites for invasive mosquitoes, which may include truck stops or distribution centres that receive a lot of cross-channel traffic. Service stations are important hubs for the movement of cross-channel traffic, and ever since Aedes albopictus was established in Italy in the 90s, as the species became more established in Italy, it moved into neighbouring countries, in the majority of cases through the movement of vehicles. And we're particularly interested in the movement of goods vehicles because they travel long distances in fairly short spaces of time. And this works because adult mosquitoes will hitch a ride in the cab or in the goods part of the vehicles. And then when the vehicle stops, the mosquito will get out and perhaps seek a blood meal, or perhaps be seeking habitat in order to lay eggs. So the OV trap is a very simple trap. It's particularly suited to Aedes albopictus because it mimics a tree hole habitat that this species in its native habitat would be particularly focused on. These simple traps will fill with water and place the polystyrene in, and the mosquito will be attracted to the OV trap in order to lay its eggs on the polystyrene. We found Aedes albopictus a number of times over the last few years, and working with a local authority, it's really important there's been a rapid control response, and that has consisted of the removal of potential habitat in the local area, on site and within a few hundred metres of it. These mosquitoes only fly a very short distance as an adult mosquito, and so we can focus the control efforts very much on that area, removing litter, removing other rain-filled habitat that's suitable for this species, and ensuring through the use of lots more mosquito traps that there aren't any other examples of these species in that location.
Back in 2016, there was the emergence of Zika virus in the Americas. And one of the key questions we had at a government level was whether we had the right vectors for this emerging virus to cause local cases here in the UK. Now, prior to that, we'd already established invasive mosquito surveillance in 2010. So we were already able to say that through our surveillance at seaports, airports, motorway service stations, inland border facilities, that we knew that we didn't have these mosquitoes currently in the UK, and therefore the risk was low. Since then, we've been monitoring the mosquito. We found it on 6 occasions coming in to these points of entry at truck stops on highway stations, and we now have a program whereby we're monitoring both the mosquitoes in the adult traps and also mosquitoes laying eggs in these service stations. And we have a contingency plan where we work with local authorities, health protection teams, where we can rapidly identify that we've got the mosquitoes, implement a control plan with the local authority, and prevent establishment of the mosquito.
So we have a network of traps as part of our nationwide mosquito project. And those traps will run from April to October, which is the majority of when we might expect the majority of species to be active in the adult stage. And the data will then help us understand key seasonality of these species, when we can expect adult mosquitoes to be biting or to be active, because that information is crucial to understand their role in the cycle of mosquito-borne disease, whether that's West Nile virus, which is primarily a virus of birds, or other viruses that we may be interested in. This is a mosquito magnet trap, and it's designed to attract mammal-biting mosquitoes. And we use these traps at a range of sites across the country to understand the seasonality and distribution of our native British mosquitoes. The trap produces a plume of CO2 using this propane cylinder, plume of CO2 and heat, and also has a pheromone lure inside the trap called octanol. And together it produces the cues for mammal biting species. And in this way we can understand the seasonality and distribution of key virus vectors such as West Nile virus and where they're found. In fact, at this site in Thames Estuary we found Culex modestus, which is potentially an important species for West Nile virus transmission.
West Nile virus causes encephalitic disease in humans and West Nile fever, and caused a large outbreak in the United States in 1999, and has since then spread right across the United States. Has now emerged in Europe, and through the trapping that we do and the larval sampling, we can identify where the key vectors are, take those mosquitoes back to the lab and test them for arboviruses.
Tipping for larvae allows us to associate a particular mosquito species with their specific aquatic habitat. And that's very useful information when it comes to understanding the ecology of that species in the environment, understanding where it's found in the landscape, understanding which habitats support certain species. And potentially one day it could be useful information in order to target mosquito control, very specifically targeting particular species that we're interested in from a vector-borne disease perspective. When we bring in mosquitoes from the field, we may be doing so from the mosquito trap, in which case the mosquitoes are likely to be dead, and we want to keep them at cold temperatures so that we can identify them on dry ice, keeping them cold, and then we can take them to our colleagues in the virology department to test for viruses. Welcome to our rearing room. So we keep the door closed because we have live adult mosquitoes in here. And we have them in here for a number of reasons. You can see some live mosquitoes in here flying around the bug dorm. If we have invasive mosquitoes, we'll bring in eggs into one of these bug dorms and we'll soak them so the eggs hatch into larvae and then grow into adult mosquitoes. And we'll do that so we can have morphological ID of all the different life stages. We also have the facilities to blood feed the adult mosquitoes, so we can provide a blood meal, and that will enable the female mosquitoes to lay eggs to colonise that population, that species. And later on this year, we've got some work planned where we're going to challenge some of our native species with different larval control products and see how those products affect those species. One of the questions we are interested in is whether we have the right mosquito control products targeting mosquito larvae available in the UK and whether they work against British species. This will allow us to be prepared to control mosquitoes in the event of mosquito-borne disease. The pathogens that we're working on in terms of these vector-borne diseases that we're interested in are very sensitive to climate change. The Medical Entomology Group now sits within the Centre for Climate and Health Security in the UKHSA. And that's really ideal for us because Over the coming years, climate change may impact upon the occurrence of vector-borne disease in the UK. You just need to give it a good clap.
Hi, hello. I'm Professor Leah Barang-Ford, and I head the Centre for Climate and Health Security at the UK Health Security Agency. The UKHSA established the Centre for Climate and Health Security to bring together existing expertise and build new expertise to enable the UK government, local governments, and partners to prepare for and build resilience in response to the threats to health that are posed by climate change. The work that we're doing on mosquito and tick surveillance is absolutely critical because we need to understand exactly where and when new species of mosquito, tick, or the pathogens that they carry are emerging in the UK, because that will allow us to deploy rapid response to minimize those populations and either prevent them from establishing in the UK or slow their spread. And that will have important implications for minimizing the risk of new and emergent pathogens within the UK.
We know that in Europe, West Nile virus and Usutu cases are found each year, particularly in recent years. And it's likely that with climate change, that those viruses are going to be detected more often in the UK. So it's really important that our work conducts a surveillance for both the species that are going to be of most importance for vectoring those pathogens, but also we work with virologists to detect those pathogens within mosquitoes.
Ticks have actually been around for a really long time. There are actually some reports of them being found in amber back from when the dinosaurs were walking on the planet. This just goes to show that they're really good at evolving, and this is something that we really need to keep an eye on, particularly with future climate change scenarios. Ticks are not going to go away. They're going to evolve to the new situations and the changes that we make to our planet. And one of the challenges here is going to be scaling up this sort of regional response that we have to these incidents that might be arising. So the more people that engage in the surveillance schemes and the more people that engage with UKHSA on their local issues, feeding us information so that we can feed information back, the better.
From an entomology perspective, the policy decisions we make today will have implications for vector-borne disease risk in the future. This includes, for example, the ways in which we decide to green the UK under decarbonisation. So many decarbonisation strategies involve nature-based solutions or greening different landscapes. And it's important that we do so in a way that doesn't create new habitat for vector-borne disease and generate new health risks. Secondly, we need to decide what our approach will be to controlling vector-borne disease and mosquito risk in the UK. It is possible to prevent or dramatically slow the invasion of mosquitoes and new pathogens, but that takes huge investment in surveillance, in mosquito chasing, in mosquito testing. So the decisions we make today will affect the extent to which new diseases become established in the country in the future.
One of the important aspects for the future is to raise awareness amongst all of us of the threats posed by vector-borne disease. I've worked in the field for 25 years, been exposed to lots of ticks and lots of tick habitats, but I'm aware of how I can minimise my risks associated with tick-borne infections. So it's all about passing on that information to the public and also our future generation.
Climate change cannot be reversed. We have to learn to live with it. And importantly, to minimize the level of warming, because that will minimize our risk, and then to adapt to the risks that we've already committed to through past emissions. The last generation committed us to a level of impacts that we cannot avoid. What we can do is avoid and minimize and influence the impacts of the next generation.